Hi, everyone. It's my great pleasure to introduce our first speaker, Carey Nadell. Carey got a bachelor degree in biological sciences, University of Oxford, PhD in ecology and evolutionary biology in Princeton University, supervised by Simon Levin. After a short postdoc with Bernie Bachelor in Princeton University and the series scientist in Max Planck Institute in Germany, he started his own group in Dartmouth College and Department of Biological Sciences in 2018. Also, Carey was awarded a lot of grants from NIH and NSF. He served as a peer reviewer of many journals. Carey's research focused on the bacterial structure and organization in community level. He did excellent research on mechanism of biofilm matrix and the bacteriophage interaction in biofilm environment. Without further ado, please welcome Carey.
Thanks so much for the introduction, and thanks for the community here for inviting us. This is just such a pleasure. I can't wait to hear all of the amazing speakers who are here. And of course, after our hiatus from visiting places to talk to people, like each one of these events is like even more inspirational and fun than normal. So I'm so happy to be here, and thank you for inviting me to participate. Can I just check that everyone can hear me in the back? Great. This is a great room. Okay, so without any delay, I'm going to get into things here. So I'm showing you a video now of a couple of cell lineages of Vibrio cholera. They're on a piece of glass under flow of nutrient media. So again, this is a group of bacteria that are forming a multicellular biofilm architecture. We often call these biofilms in microbiology. And their ability to do this depends on the secretion of often a pretty complicated array of adhesives, usually polysaccharides, proteins, some DNA maybe. And these adhesive compounds have to interact with each other in a specific way to achieve the arrangement of cells that you see here. And this arrangement has an important consequence for how these groups interact with other pieces of their ecology, including predators, which is what I'm going to talk about. So in keeping with the theme of this symposium, I want us to be thinking about how cellular arrangements in group like this influences microbial ecology, and how we can think about the behavior at cellular scale influences the ecology of big groups of cells at multicellular scales, like millimeter scales in my case here, but like maybe even larger than that. All right, so before I get into the details, I just want to make a note. It's a little redundant with the introduction, but my background is in theoretical ecology and evolution. So moving into molecular biology was fun and difficult. The pace of changing the color of bacteria, for example, is way slower than if you're doing a computer simulation. But eventually we got used to that, and so we tried to combine perspectives from ecology and evolution and with some mechanistic insight from molecular microbiology to understand fundamentally ecological questions, but from a range of different scale perspectives. So we do some work that's more applied. So we work with people at the hospital at Dartmouth to understand biofilm growth, for example, on titanium and other metallic surfaces used in prosthetic implants and hip and knee replacements, for example. But most of our work is basic in comparison, and we have a particular focus at the moment on predation in biofilms by viruses and by other bacteria. The main reason is because it's cool and it's fun to study, and the second reason is because there's very little work on this. It's difficult to visualize, and so we spent a lot of time making tools to look at this and see what is happening. So the main model I'm going to be talking about today, there's some others, but the main model is Vibrio cholera, which is famous, of course, for causing the disease cholera. It's actually only a minority of cholera strains that can do that, though. Most are just marine microbes. They grow in the ocean and estuarine conditions, and they colonize particles of chitin and alginates that are floating in the water column and eat them. And they eat them by forming biofilms on their surface and secreting enzymes that digest them into smaller components that they can take up for biomass production. So biofilm formation is an important part of the ecology outside of host. That we think it also contributes to pathogenesis, but I'm not really going to talk about that. The way that we cultivate them, we do it in a couple of different ways, but we mostly use microfluidic devices, like what I'm showing here on the right. Well, I was told to point with this thing. I'll point with this thing. These devices contain basically just simple tunnels, like 1, 2, 3, 4, 5 tunnels here. So we inoculate those with cells on glass or on particles of chitin sometimes, and then flow media over them and allow them to grow up. And we image them at very high resolution using confocal microscopy. So almost all of our raw data are images, confocal images. All right, so what is our predator here? So the predator, and this is really fun, I think, is another bacterium called Vibrio bacteriovorus. Not related to Vibrio cholera at all. It's confusing in the genus name. That just refers to the shape of the cells. That refers to the curved shape that they have. This bacterium behaves a lot like a phage, like a lytic phage. It actually finds hosts. It pokes a hole in their outer membrane. It actually pokes a hole through their cell wall, which is then patched. And then they live in the periplasm of the host. They secrete a bunch of digestive enzymes into the cytoplasmic compartment and dissolve everything, and then they absorb what is released. They grow into a filament, and they eventually divide according to however many times they could copy their chromosome. So these guys are very common in the natural, in the wild. So they've been found associated with marine snow, which is another word for those chitin alginate particles that cholera colonizes. When there's a lot of it, it looks kind of like snow falling in the ocean. It's just bacteria, though. It's not. It's not actually so. They're found on bivalve shells, in freshwater, in soil, and sewage, in fouling biofilms. So these are ones that are found on surfaces where we don't want them to be, like on inside pipes or on ship hulls. Plant surfaces, animal guts. These bugs and their relatives are quite easy to find, in short, and they have a very broad host range of gram-negative prey. And this is what allows them to compete with phages, which reproduce a lot faster than they can, but usually have a much narrower host range. So we got interested in these talking to Daniel Kaduri at Rutgers. And because they hadn't been visualized before at high resolution in biofilms, we just didn't know what they look like, what their activity is like in there. So like many of our projects, we started by just looking and waiting to see what happens. So I'll start in with the image data now. So if we grow a biofilm of cholera, which is going to be in red, and then we introduce Vibrio, which is going to be in cyan, we see this. And obviously there is some successful predation. You can see that in the overlap of the predators and the prey. They also round up, which is called a delaplast, as the host cell wall is modified. So we see predation. So the Vibrio can kill cholera, and we see that happening around the periphery here, but there seems to be a protected zone in the middle of this structure, and that is stable over time. So 2 days later, this colony has grown into this structure, and there's still some predation perhaps happening, but a lot of the Vibrio has been kind of immobilized, and the group maintains positive net growth. So they are protected overall. Okay, so how is that? What is the controlling factor here? We naturally look to the biofilm matrix, that complex of secreted compounds that they make that I mentioned. We found in the past that the matrix itself can block phage exposure. So we thought maybe the matrix was blocking Vibrio exposure. And so I'm showing you an image like the one from before, but now the matrix is labeled 2. That's the yellow part. We're labeling a protein called RBMA, which is one of the primary matrix components for cholera. It's distributed fairly evenly throughout, which makes it a good proxy. So if we look in more detail at this picture, we can see a couple of different things happening. So we can see isolated cells, which in general are not in great shape. They're mostly dead. There are some that aren't found, though. And then there are different size groups of cells that are making matrix. There's smaller ones and there's large ones. And it seems like the smaller ones can still be killed. And on the right here, I'm showing something we call the degree of predation, which is a measurement of overlap of the prey with the predators. So they're being killed, which suggests the matrix by itself is not enough to protect them. It's really only the middle of these groups that are larger that seem to be protected. So there's something more subtle than just matrix protection going on here. So I'm going to run through some of the type of quantification we do. We place a large emphasis on being as precisely quantitative as possible, rather than just looking at the images and hoping that our intuition is correct, basically, after looking at them. The way that we do this is by segmenting the 3D image stacks. So we separate biomass from backgrounds. And this is the same image actually I showed you in the last slide, except looking laterally after rendering in 3 dimensions. And then after we do that, we chop the whole thing into a 3-dimensional grid. And this is kind of like capturing either single cells or small groups of cells in the biofilms. And then we can go to each of those cubes and analyze the properties locally, so we get a spatially resolved measurement of various things, quite flexible. So in this case, we're interested in a couple of different biofilm features, like the packing architecture. So what is the volume fraction of biomass in close proximity to a chunk of cell? Or what is the volume fraction, what fraction of this volume in a larger neighborhood is filled with other cells? This is that degree of predation, so the overlap of predators and prey. And then this is the amount of matrix in proximity to cells. So these are the 4 measurements I'm going to be talking about here. And I'm not sure, is it allowed for people to ask questions in the middle of the talks?
Yes.
Okay, if anything is unclear, please ask now rather than later. So I'm showing you 3 different prey strains. So the left is wild type. That's what I've been showing you already of cholera. The yellow is still the matrix stain. This is a hypermatrix secreting strain. So you can see more yellow stain here. And then this is a mutant that's missing RBMA. And RBMA is really important for holding cells in close proximity. So you can see already that they're much more spread out, and it doesn't seem to be good for them with respect to the predator's activity. So I'm going to plot here the local biovolume fraction, so that small scale packing, and then the neighborhood volume fraction, so the larger scale packing. And these are all the cubes that we dissected out of many, many of these image sets that we took. And they're color coded according to that predation measurement. And we can see already, let's just look at wild type here, that the local biovolume fraction doesn't predict predation that well. There are black dots all throughout this x-axis. On the other hand, the neighborhood packing fraction does a much better job, especially above this kind of 0.8 threshold. And that we see that again with this hypersecreting strain, but most of the cubes are up there because it's making more matrix and they're more packed together. The mutants, on the other hand, is faring quite poorly, and it never reaches that packing fraction on the large scale, and most of them are dead. Okay, so we can, if we can analyze these data formally, statistically, and in fact, local biovolume fraction doesn't predict predation susceptibility at all, whereas neighborhood does. These are the same data for the 2 strains that are making matrix. Now the color scheme is with matrix production, and this is just to show that this strain has much higher matrix production on average, but it's still that neighborhood packing that really controls protection here. So I'm kind of going into the weeds here with the analysis just to emphasize that by being careful with the way we quantify these image data, we can pick out details about what is controlling the ecological interaction here. The matrix is important because it is controlling architecture, but it's not the matrix by itself that blocks predator access. It's the architecture, the arrangement that it confers. I hope that's clear. Okay. So this has some other consequences at larger scale. So these are 2 biofilms, again, rendered in 3 dimensions, and they're locally colored according to that neighborhood volume fraction. We also have started calling this cell packing just because it's more intuitive. So I might say that sometimes. This is in the absence of predation, and this is when predation has occurred. And you can see that the biomass distribution has shifted dramatically, because when predators arrive, they kill everything below that critical packing, and anything that's above remains and has more space to grow and expand. So the landscape, so to say, at this microscale landscape has changed quite dramatically as a result of the predator activity, which again, we can quantify precisely, but I'm going to skip that part because it's the images tell the story. The other thing that this changes is the way that the residence biofilm population can interact with planktonic populations that are in the vicinity. So my last postdoc project with Bonnie Bassler was showing that existing biofilms are pretty difficult to invade by planktonic cells. It's almost as though they're kind of protecting the space that they have. And this is an example of that. So this red biofilm was grown for a couple of days, and then we threw on a yellow population, actually of the same strain, and they don't really colonize that well, and they don't get into the middle of the group. But if there's a predator there first, it is able to kind of chomp around the periphery of these groups, which don't have the same packing fraction as the center, and that loosens the architecture to the point that other cells can attach again as well. So this, from an ecological perspective, we would say that this can alter the way that succession works, or alter the strength of priority effects in a given location. And by that I mean that in ecology we say there's a priority effects if whatever species arrives in a new resource patch first gets it, and whichever one it happens to be that arrives first, that's more or less. So that we see that kind of effect with biofilm behavior quite a bit, but in this case, it's altered by predator activity. Okay, and this is true like regardless of what species you throw in there.
Can I ask a naive question?
Please.
Are the Vibrio or the predator, are they motile?
Very. Yeah, okay. They're very fast.
And so they can't penetrate into the path matrix, but they move around the.
Yeah, okay. and they can kill cells along the periphery, where the group is expanding and they haven't kind of consolidated that architecture yet, so they're not packed enough to prevent their access. I mean, I will admit that there is something still missing, because even on the boundary of where the packing is sufficient, why can't they just kill the ones right on the outside and then work their way in? I'm not sure. There's something, I'm not sure. Yeah. Okay, so we see that this protection effect depends on architecture in cholera. Is that true are other species able to protect themselves from Vibrio predation in that way? So the answer is no. I imagine some can, but E. coli can't. And so we did the same experiment here with E. coli in yellow and Vibrio again in blue. This is a slightly different visualization. Like the central images in these panels are XY slices, so kind of a lateral slice through the biofilm, and then this is a vertical projection, so a vertical slice. And you can see in this case, the predators just chew their way all the way down through the biofilm to the surface. So these E. coli doesn't protect itself in this way, even if their biofilms are relatively as mature as we can get them in our experimental context. This is just a quantitative confirmation that the host is gone at the end of this experiment. They are dead, all of them. Yes? Is this a natural predator of E. coli? We have no reason to doubt it, because we can find they can be found in the same places that E. coli can be found. I'm not aware of so it's a little bit inferred, but we don't have reason to doubt it. Okay, so we have one prey that can protect itself with its organization, its architecture, and another one that cannot. So naturally, we were curious what happens in a multi-prey context if both prey are growing together, with one can protect itself on its own and the other can't. What happens? Okay, so I'm going to show you some raw image data first. These again are lateral optical sections, so just a 2D slice through a 3D system. And then these are subpanels. These aren't vertical projections anymore. They're just smaller, kind of higher magnification images of what's going on. So it's complicated, right? It's a little bit hard to say. So we'll exactly what's going on here. So we need to start with some broad quantitative characterization. So
which cell were in which color?
Oh, I'm sorry. So cholera is in red still, and E. coli is in yellow. Thank you. And again, and the predator is in blue again. So let's look on the left here. So the solid lines, red is cholera, yellow is E. coli. In the solid lines, we have the monoculture conditions. So in monoculture, so cholera is by itself, it's growing along, the predators arrive, and they don't really care that much. Or their growth is reduced, but not that dramatically compared to the absence. The absence data are not being shown here. E. coli, on the other hand, is just wiped out completely. They're gone when the predators arrive. So when we look at the dual culture condition, which are the dotted lines, we can see that E. coli does much better than when it's on its own, and cholera does worse, right? And this is just so this is on a log scale, so we can see everything. But looking at this another way, cholera in monoculture actually keeps on growing on net after predator exposure. This is a percent bivolume change, which is population size change from here to just before predation, so 48 hours after predation. So cholera has continued to grow, but when it's with E. coli, it's still below where it was. Whereas E. coli is just gone when it's on its own against predators, but there's some of them do survive when they're co-cultured with cholera. So why is this happening? This is quite interesting, this pattern, especially to a social evolutionist, someone interested in cooperation and conflict, where you put two things together and one does better and one does worse relative to when they're on their own. Let's think about this one first. This is a bit more intuitive. So if you look at these data, if you look in these images here, you can probably get a sense of what's going on. You can see some E. coli inside of these dense cholera structures, and they seem to be avoiding exposure to predators. And that is what's happening. So at least some fraction of the E. coli population gets trapped inside of the cholera population, and they basically reap the benefits of the architecture that cholera is producing. They are trapped in there, but they're still viable, and I'll show you that in a second. This is just a formalization of the image data here, but these red lines are the borders of the cholera groups, and the pixels are E. coli coded by whether or not they're being eaten, and the ones inside the groups obviously aren't, whereas the ones outside are. And then this is how much cholera is around the E. coli, this measurement. And then if we relate those to each other, we get this nice quantification showing that with enough cholera in proximity to E. coli, which is this measurement on the x-axis, the E. coli is protected. Okay, so that results is pretty straightforward. And again, the E. coli in there are viable. They're not just being smothered and starved out. So this is a later stage image of the same thing, much larger spatial scale this time. And if we remove this with very fast flow, so like very large shear stress, and recolonize a new chamber later, the E. coli is still around. So they're still viable. They're not winning this competition by any means, but they're doing better than they would if they were by themselves. Okay. So I'll just add really quickly here that this protection mechanism is not unique to Dellavibrio predation. This is another example where we're using lambda phage against E. coli instead of Dellavibrio, and we see the same exact thing. So E. coli trapped inside these cholera colonies are protected from lambda infection, and the ones outside are not. So this graph is kind of another. It's another version of the same one I showed you in the previous slides, the same fundamental shape. Okay. We okay on time? Yeah, great. So now this part of the result, why is this happening? Why is cholera doing worse when it's co-culture with E. coli and they're exposed to predation? This one's trickier. So this is an example of what we do in my lab a lot. We take a lot of images and just stare at them for a long time and try to get a sense of what might be happening, and then we start to measure things to see if that corresponds to our intuition, because ultimately your brain is a better image processing device than any computer is for now. So we spend a lot of time just trying to understand what's going on, intuitively, and you can start to see here that I've shown you what we call a conventional or normal cholera architecture that's pretty predictable. It has clusters of cells that have a radial orientation with respect to the center point of the cluster, and then they pack more and more over time. And that does seem to be interrupted in some of the locations here in this image. Yes?
Does E. coli need any genes in order to take advantage of this protective possibilities of cholera, or is it just completely passive that they're
We're not sure yet. Thank you for the question. We're not sure yet. We don't think it's entirely passive. I allude to that a little bit at the end. So I don't think this would happen with just any other species, something we're actively trying to figure out. Okay, so let's look at this case, which is a bit more it's a bit clearer. So this is that conventional cholera architecture that I talked about. And then this is this other possibility that we see where the two are mixed together. They're very well mixed. And you can see that the cell packing is much lower than it is when cholera is on its own. And the consequences for predator access are this. So in these groups, the predators are able to access everything and kill everything. Whereas here we see the protection mechanism that I described in the first part of the talk. Okay, and then this is just a quantification of the effects that we're seeing, where E. coli is mixed up with cholera in this case. That's what the color map is showing. And that's where we see a lot of predation, which is what this color map is showing. Whereas here, the center of the colony is protected, as we've seen before. So how do these two structures arise? Like they're really two different trajectories that the system follows. How are these different? So the way that we tried to pick this apart, and I wouldn't say that we've done it completely, but we've at least gotten a start, is to grow many samples and then look for the two different outcomes and then kind of trace them back in time to where they started from. Okay, and you can already see that these two seem to be rather different at the beginning of the experiment in terms of the proximity of the two species to each other. So the other thing that's different is that so like I said before, the internal part of these conventional cholera colonies has the high packing, passes the high packing threshold that it confers protection, and the other ones don't. That just never happens, which is why this whole effect occurs. So we see that this is what I'm plotting here is the cell packing or neighborhood volume fraction in the center of a developing cluster versus the periphery, so the gray bars. And we see at around the 20 to 26 hour mark in this particular set of conditions that center high packing region kind of nucleates and expands. And that just never happens in the mixed species colonies, which start like this. So if we measure the average distance between cholera and E. coli cells, we can see that these two types of structures start and remain on different trajectories throughout the experiment. So what we think controls this outcome is the proximity of E. coli to cholera on a local basis at the beginning of the experiment, or in other words, the colonization conditions, the initial population density on the surface. And then this has big effects on the subsequent population dynamics because of the influence that cell arrangement has on structure and organization throughout the growth. Okay. So just starting to wrap up here, from a social evolution perspective, that's where I kind of came from in evolutionary biology. This has prompted us to think about the architecture that cholera produces or that biofilms produce in general as a kind of public good or a cooperative product that in this case is shared among clone mates, and that's what ultimately stabilizes a lot of cooperative behaviors. And it's tolerant to some degree of mixing with other species that aren't producing the same matrix components. But if there's enough mixture between the two, then that structure breaks down, or it never really develops in the first place, to be precise. And then this has effects on the larger scale ecology of these species in the context of predation in this case. And so our lab is interested in all sorts of examples where architecture at this scale influences some fundamental ecological process at this scale and larger scales. So in the future, we're looking to move into, and I kind of forgot to add this caveat at the beginning. Cholera and E. coli are maybe not the most naturally realistic partners. I don't think it's totally unrealistic. They are found in water supplies, like near humans, as you might imagine. They've both been isolated from biofilms on the hulls of ships in proximity to humans, for example. Like it's not absurd, but it's also probably not the most common thing. We explored it just to establish the principle of how these architecture effects can be important. But we want to move in the future into more realistic types of conditions. So we're going to think about vibrios on chitin in seawater. These are two different species of vibrio, cholera on the left, Parahemolyticus on the right. And then this is an image of the two together on chitin with Delavibrio thrown in, for good measure in this case, in magenta. And so our future directions are in keeping with these themes, but with more ecological realism. So that's that's the end of my talk. So I want to thank my group, who I feel very, very so lucky to work with every day. There's this brilliant, enthusiastic scientists, our funding, of course, and our collaborators at Memphis and Rutgers for these projects. So Ben Wooker did a lot of the data collection and analysis that you saw here, but there's also some from Alice Goldstein-Plesser, who's here, and Jay Winans, who's right there. And we're, of course, very grateful for our funding sources that allow us to do all of this. So I think I will stop there. Thank you for your attention. I'm happy to answer questions.